Do you remember what the pyruvate dehydrogenase complex does? The pyruvate dehydrogenase complex links glycolysis in the cytoplasm to the TCA cycle in the mitochondria. The net reaction converts pyruvate, NAD+, and coenzyme A into three products: acetyl-CoA, carbon dioxide, and NADH. Here, we can see pyruvate dehydrogenase creating an activated carrier, an NADH molecule, when it converts a pyruvate into an acetyl-CoA. There are 5 cofactors that pyruvate dehydrogenase complex needs to function. Pyrophosphate, which is basically 2 inorganic phosphate groups, is supplied by B1 in the form of thiamine pyrophosphate. FAD, NAD+, and coenzyme A come from vitamins B2, B3, and B5, respectively. As a side note, the last one, lipoic acid, is inhibited by arsenic. If you're reading along in first aid, you'll see that arsenic poisoning causes vomiting, rice water stools, and garlic breath. That's all fine and dandy, and relatively high yield, but do y'all remember the mechanism of this toxicity that we went over earlier? Hopefully, you remember that arsenic inhibits the pyruvate dehydrogenase complex. If you're exercising, you probably want to be undergoing aerobic metabolism, and therefore, you want the pyruvate dehydrogenase complex to be active. The body's main goal will be linking glycolysis to the Krebs cycle using the pyruvate dehydrogenase complex. During exercise, the ratio of NAD+ to NADH increases since you're consuming all of your NAD+ during glycolysis to make a more reduced form, NADH, which will go to the mitochondria to make lots of ATP in the process of oxidative phosphorylation. Other changes during exercise include ATP, NADH, and acetyl-CoA, since these are all things that are produced downstream of this enzyme. #negative feedback. Where can we see a similar enzyme to the pyruvate dehydrogenase complex? The alpha-ketoglutarate dehydrogenase complex converts alpha-ketoglutarate to succinyl-CoA in the TCA cycle and uses the exact same set of cofactors as the pyruvate dehydrogenase complex. If you can remember one of these, you can remember the other ones as well, since they both use the same cofactors and they're both dehydrogenase complexes which decarboxylate their substrates. Let's talk about the clinical scenario regarding this enzyme. Pyruvate dehydrogenase deficiency is a serious problem for all cells with mitochondria, since it prevents the products of glycolysis from entering the TCA cycle, also preventing aerobic respiration. The pyruvate from glycolysis normally gets converted into acetyl-CoA from the pyruvate dehydrogenase complex. If this pyruvate can't get converted to acetyl-CoA, it'll get shunted into anaerobic metabolism to form lactate. Pyruvate dehydrogenase deficiency can be congenital, meaning people can be born without this enzyme or with a defective copy of this enzyme, or the deficiency can be acquired later in life. Why do you suspect alcoholics can present with an acquired pyruvate dehydrogenase deficiency? Because alcoholics typically are nutrient deficient, especially in vitamin B1, or thiamine, which is listed as a cofactor for pyruvate dehydrogenase. Boards love testing this concept, so don't be surprised if you see this on test day. Clinically, a pyruvate dehydrogenase deficiency manifests as neurological defects or lactic acidosis, and an increase in serum alanine during infancy. It's treated by increasing the intake of ketogenic nutrients, such as a high-fat diet or a diet high in the ketogenic amino acids leucine and lysine. But why a diet high in leucine and lysine? These nutrients do not have to go through a pyruvate intermediate to enter the TCA cycle, so they actually bypass pyruvate dehydrogenase complex altogether. We spent all that time talking about glycolysis. An endpoint of glycolysis is to make 2 molecules of pyruvate. Pyruvate is an important intersection point for several metabolic pathways. What are the different pathways a single pyruvate molecule can take? First, pyruvate can be converted to alanine in the liver via alanine transaminase, or ALT. This is how amino groups are transported from the muscle to the liver. Second, pyruvate can be converted to oxaloacetate via pyruvate carboxylase to either replenish the TCA cycle or for gluconeogenesis. Third, on our last slide, we said pyruvate can link glycolysis to the TCA cycle via pyruvate dehydrogenase. And fourth, pyruvate itself can basically be the endpoint for energy production in anaerobic glycolysis, when it's converted to lactate by lactate dehydrogenase. This reaction regenerates NAD+, so that glycolysis can continue to produce small amounts of ATP compared to the oxidative phosphorylation that takes place in mitochondria. Where in the body do we typically see anaerobic metabolism? Anaerobic respiration is the main source of ATP in red blood cells, white blood cells, the kidney medulla, the lens in the eye, testes, and cornea. Hey, welcome back. Regarding our discussion of metabolism sites, do you remember where the Krebs cycle takes place in a cell? The TCA cycle or the Krebs cycle takes place in the mitochondria and produces the high-energy electron carriers NADH and FADH2, which are used to generate ATP in the electron transport chain. This completes the metabolism of glucose. This diagram shows the basic steps in the TCA cycle. Notice how acetyl-CoA is fed into the system and the cycle spins, creating the various activated carriers which we talked about earlier. It also produces GTP, which is the equivalent of ATP. Once the cycle's done, the TCA cycle is ready for another input of acetyl-CoA, and we'll keep on rolling. So this was a very basic example of the TCA cycle. Now we'll add in some enzymes and intermediates to spice things up. Pyruvate is the main substrate, or input into the cycle, and this is converted into acetyl-CoA by pyruvate dehydrogenase, or PDH for short. This reaction produces one NADH and one CO2. Protein and lipids can also enter the cycle after being converted to acetyl-CoA, emphasizing the fact that carbohydrate, protein, and fat metabolism are all connected at this step. The net reaction of the Krebs cycle produces 3 NADH and H+, or hydronium ions in solution, 1 FADH2, and 2 CO2, and 1 GTP per cycle, which amounts to 12 ATP per acetyl-CoA. What do these activated carriers end up? Well, these activated carriers can exchange electrons later in the electron transport chain to produce ATP. We'll see later how each NADH can be used to make about 2.5 ATP, and each FADH2 can be used to make about 1.5 ATP. Since each glucose yields 2 molecules of acetyl-CoA, this means you can get 24 ATP from each glucose. The TCA cycle is regulated at 3 major irreversible steps, which are catalyzed by the enzymes citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. FYI, you probably won't need to know anything about the molecular structures of these intermediates for the USMLE, so take a breath. Citrate synthase, which can be inhibited by high levels of ATP, combines acetyl-CoA with oxaloacetate. Citrate synthase combines acetyl-CoA and oxaloacetate to yield citrate. Isocitrate dehydrogenase converts isocitrate into alpha-ketoglutarate, yielding 1 CO2 and 1 NADH and hydronium ion. Notice that we lost a carbon there as CO2, and also notice how logical it is that this enzyme is inhibited by ATP and NADH, since if you have a lot of those, there's no need to continue going through the TCA cycle. Our last irreversible step is alpha-ketoglutarate dehydrogenase, which converts alpha-ketoglutarate into succinyl-CoA. In doing so, as I mentioned before, the alpha-ketoglutarate dehydrogenase complex uses the same cofactors as the pyruvate dehydrogenase complex. Do you remember what these cofactors are? The pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase use vitamins B1, B2, B3, B5, and lipoic acid. As a side note, do you know which important synthesis pathway starts with succinyl-CoA? That would be heme synthesis. This is an important concept. The TCA cycle is responsible for creating ATP, but it also creates intermediates that can feed into other pathways in the body. So flash quiz here. At which steps in the Krebs cycle are NADH, GTP, and FADH2 produced? This question is asking where our activated carriers are created. NADH is made at 3 points, when isocitrate is converted to alpha-ketoglutarate to succinyl-CoA, and malate to oxaloacetate. GTP, which again is equivalent to ATP, is created when succinyl-CoA is converted to succinate. FADH2 is made when succinate is converted to fumarate. Now, let's divert our attention to the mitochondria, where we'll find the electron transport chain. There are quite a few fancy figures out there, but I'll make this as simple as I can, because knowing all the minute details is not important for the boards. However, the things that I discuss will be important, so please pay attention. We start out with 5 major complexes, named complex 1, 2, 3, 4, and 5, and each has a unique function. Here, we have our inner membrane of the mitochondria, with the mitochondrial matrix, below the membrane, and the intermembrane space above the membrane. Our previously made activated carriers, like NADH and FADH2, dump their electrons at these complexes to ultimately create ATP. Where did we make these carriers? Remember we made some NADH in glycolysis using the enzyme pyruvate dehydrogenase, and also in the TCA cycle at various steps. We also made FADH2 in the TCA cycle. NADH enters at complex 1, and FADH2 enters at complex 2, which happens to be 6-8 dehydrogenase from the TCA cycle. As electrons pass through these complexes, they create a proton gradient that's used to drive the production of ATP. Complex 4 is also known as cytochrome C oxidase, and this transfers electrons to the final electron acceptor. Do you remember what that is? It's oxygen, which winds up turning into water after accepting its electrons. Without oxygen, there's no final electron acceptor, so the ETC cannot function, and the body must now rely on anaerobic metabolism. Complex 5 is also called ATP synthase. It uses the hydrogen gradient that complexes 1, 2, and 3 make to produce about 3 ATP for every NADH and about 2 ATP for every FADH2. Since FADH2 starts at a later complex in the pathway, it isn't able to pump as many electrons across the membrane, so it only makes about 2 ATP. Complex 5 takes ADP and inorganic phosphate to form high-energy ATP molecules. We can step aside now and introduce 3 scenarios where oxidative phosphorylation becomes poisoned or inhibited. The electron transport chain inhibitors, such as rotenone, cyanide, antimycin A, and carbon monoxide, directly inhibit electron movement from complex to complex. This prevents the formation of a proton gradient and therefore the synthesis of ATP. Rotenone inhibits complex 1, antimycin inhibits complex 3, and cyanide and carbon monoxide inhibit complex 4. Oligomycin is an ATP synthase inhibitor, and therefore inhibits complex 5. This allows the proton gradient to build up without creating any ATP synthesis. If you create a logjam in the electron transport chain, you'll create a buildup of NADH and FADH2. How do uncoupling agents work? Uncoupling agents, such as 2,4-dinitrophenol, once used as a weight loss agent, aspirin, and the protein thermogenin, which is produced in brown fat, all increase the permeability of the inner membrane, which allows protons to flow through. This results in increased oxygen consumption, since an increase in electron transport attempts to compensate for the permeability. Still, ATP cannot be created without the proton gradient, and the increased electron transport activity is instead dissipated as heat. This is why aspirin overdose can cause fevers, and why babies and hibernating bears can stay warm, since they have lots of brown fat. So, another flash quiz. A 4-year-old girl swallows some cyanide-containing rat poison from under the sink. First of all, for the love of Job, please keep your rat poison away from your babies. Thanks. Okay, how does this affect her ATP production? Remember that cyanide directly inhibits the electron transport chain, specifically complex 4, which decreases the proton gradient and blocks ATP synthesis. Therefore, the body isn't able to create ATP via oxidative phosphorylation. Please, lock up your rat poison, folks.